Hello, and a very good evening to all joining us for this somewhat unusual Gift of Sight meeting. This is the 7th such event. Previous 6 have been surprisingly public. Today is rather more homely. Who knows, we might get to like it. On behalf of the University of Southampton, I welcome the many staff, students, supporters, and alumni joining us from around the world. I also extend a very special welcome to Lady Mary Fagan, the President of the Gift of Sight. My name is Andy Luff. I'm an eye surgeon working locally, and the good news is I know nothing at all about science or research. I am selected because it forces our presenters to speak in plain English and use short words that even I can understand. I worked at Southampton Eye Unit for many years, and I still support the Gift of Sight. It's also marvellous to see the progress made by a world-class department now, providing excellent research and making a real impact upon eye disease. It's wonderful that you can join us this evening as we learn about sight loss, the research that fights it, and the incredible help given to our researchers by the Gift of Sight appeal. Visual loss is perhaps one of the most frightening of diagnoses to confront a patient, but work here in Southampton will mean a real difference for patients facing these decisions at their most vulnerable. Most treatments, most innovations depend upon basic medical science, and a recent example of good medical science locally is the setting up of a donor eye bank by the Gift of Sight appeal, allowing our researchers to look in microscopic detail at the very cells where disease starts. You might think that quality research is the preserve of universities in cities with dreaming spires, or maybe coiffured Californian campuses, but nothing could be further from the truth. Tonight, we have a stellar lineup, all brought to you from the University of Southampton. We will begin in a moment with Retina, who is a true scientist. We're then followed by 2 clinical scientists. These are individuals who are medically qualified and split their time between medicine and research. These individuals bridge the gap between intellectuals like Arjuna and simple plumbers like myself. Those 2 are Jay Self, who I well remember from his training in Southampton, all for good reasons, I can promise you, and I'm delighted to see how Jay's career is blossoming. And the second is Professor Andrew Lotry, a fellow consultant in Southampton. Now, after the presentations, we will move into a question and answer section of the event, and to submit questions, I have to ask you to use the Q&A box, which you can see on the right side of your screen, and we hope to get through as many questions as possible. So without further ado, I'm going to turn to Dr. Arjuna Ratnayaka, who by trade is a cell biologist, but now working largely in the field of eye research. Arjuna, over to you.
Hello, I'm Arjuna Ratnayaka, a lecturer in vision science. I'm interested in understanding how retinal diseases develops and the links the retina has with the aging eye. In this talk, I will be presenting some data showing how an unhealthy high-fat diet causes damage to our retinas. Sight loss sits at the center of 3 things: aging, which is the greatest risk factor, genetic, as well as lifestyle risk factors. And it is the last thing, the lifestyle risk factor I want to talk about and look at the effect of diet on diseases such as AMD. So it's known that consuming a healthy, a balanced diet can offer a measure of protection against diseases such as AMD, in the same way that consuming an unhealthy high-fat diet, termed a Western-style diet, can increase the odds of developing sight loss in later life. And there is a lot of evidence that has been published looking at large populations of humans to come to these conclusions. A group of compounds that's worth mentioning here are the macular carotenoids that also offers a measure of protection against retinal damage. But in this talk, I will not be focusing on macular carotenoids, but instead we'll focus on the effect of high-fat diet. So one of the questions I would like to pose to you is, where in the nutritional spectrum is your diet? A typical American European diet has a very high fat content. And we know that more than half of the UK population is either overweight or, in fact, obese. So it's important to understand this diet, the relationship between diet and the sight loss. So we've done that using mice. And this is a study that was undertaken by Dr. Ellie Keeling and Dr. Savannah Ling over a number of years, where we used common garden variety mice. So these are not genetically modified in any way, and that are maintained to very high standards in our laboratories. These rodents are given a normal diet, which typically contains 7 to 10% of fat. Now, scientists who have studied the effect of obesity give mice a high fat content diet ranging from 25 to 60% of kilocalorie fat. We used something in between, 45% kilocalorie fat. With these diets, the mice gain weight, become obese. They develop metabolic disorders as well as sleeping problems. The 45% kilocalorie fat diet is considered by experts to be closest to modeling the Western style diet in humans. And we've done this, we've given this diet to mice over a 12-month period. Now, I want to take you to the back of the eye, to the retina, and look at the different layers that make up the retina. If you look at the retina in cross-section, it's very much like the different layers of a cake, like this, where light comes through the top part and focuses on the photoreceptors, a layer of cells that enables us to see, which sits pretty much towards the bottom end. So here's a cartoon I've drawn for you, and I've coded all the different layers of the retina, and it's just like a layer of cake. Light comes through the top part to focus on photoreceptors. These are looked after by a carpet of cells called the RPE layer, or retinal pigment epithelial layer. If RPE cells become damaged and dysfunctional, they become unable to look after the overlying photoreceptors, which means they also die, which is when you develop macular disease. The RPE sits on a thin porous tissue called the Brooks membrane. And underneath, you have the blood vessels called the choroid, which supplies oxygen and nutrients from our diet to these tissues, and then takes away the enormous metabolic waste that's generated. It's worth noting here that with age and onset of disease, such as AMD, we find the appearance of fatty deposits called drusen underneath the RPE. So the inner retina is where the light comes through to focus on the tissues of the outer retina and photoreceptors, and it is the tissues of the outer retina where we see most changes in AMD. I'm going to come back to this slide, this cartoon, time and time again now, as we go through our results. So here we are looking at a tissue, a cross-section of the retina from a mouse fed a normal diet, and compared that to a mouse fed a high-fat diet. We used beta-3 tubulin, which is labeled in green. It labels up all the neurons in the retina. And we looked at the fluorescence in each layer. And we found that the fluorescence in the retinal ganglion cell layer in mice fed a high-fat diet was significantly reduced compared to mice fed a normal diet. So they appear to have fewer retinal ganglion cell processes. And I will talk about the impact of that later. We also looked at a protein called clusterin, which collects in the RPE Brooks membrane here, comparing between the control diet and the high-fat diet. And we find that the clusterin levels were increased in mice with a high-fat diet. Clusterin proteins are linked with AMD. Another protein we looked at is TIM3, again in the same layers, and we find high levels of TIM3 in the eyes of mice with a high-fat diet. TIM3 proteins are linked with aging, AMD, and a rare blinding disease called Salisbury's disease. We then look to see if lipids or fats actually collect in the eyes of high-fat mice, and we see in red lipid or fat droplets in the outer retina of these mice, and hardly any in mice fed a control diet. Then we used another type of microscopy called electron microscopy to peer even deeper into these tissues. Now, these enable us to magnify these images about 3,000 times. And here you can see an electron micrograph from a mouse fed a normal diet. I've color-coded the RPE for you, the Brooks membrane in blue. And if you orient that to the cartoon, you will see where all the tissues are. These are the outer segments of photoreceptors which come down. So these structures. Outer segments are gobbled up by RPE cells every day when we open our eyes. And that's important for our normal vision. And you can see the beautiful arrangement of these outer segments in this mouse fed a normal diet. Now, compare that to what we see in a mouse fed a high-fat diet. I've color-coded the RPE here in dots. Showing the RPE, you can see the Brooks membrane looks abnormal, and you can see the outer segments look very disorganized. We see evidence of RPE cells leaving this carpet of RPE monolayer in a mouse with a high-fat diet. This is what you see in AMD patients as well. And here we see a high-powered image of a lipid droplet that we saw earlier. Now, I've coded the RPE here in dots to show where it is. And in this image, we see in this high-fat diet-fed mouse, the appearance of drusen, these lipid droplets underneath the RPE, which you can see in yellow. We see abnormalities in the Brooks membrane of these high-fat diet mice and also abnormal blood vessels. And looking closely into the Brooks membrane, I've color-coded the RPE here, you can see the Brooks membrane delaminating, much like a tire coming apart in a car at high speeds. So let's summarize our findings. We found fewer or damaged neuronal processes in these mice fed a high-fat diet. And of course, the axons of retinal ganglion cells, which we saw, are the ones that actually make up the optic nerve. We see fat droplets in the outer retinas of these mice. We see high levels of AMD-linked proteins collecting under the RPE. And we see that the fine structure in all of the layers have changed, very much like you find in early AMD. So the take-home message is that we show that feeding an unhealthy diet and an unhealthy diet alone is enough to bring about significant changes to the retina. And you have to consider that in the context of genetic risk factors as well as aging. I hope you enjoyed that presentation, and I will leave it at that. And I would like to thank my team, my colleagues at Southampton, and to you for your attention. Thank you.
Arjuna, thank you very much. That was wonderful. I have two immediate observations. The first is that I'm now extremely hungry. And the second is I have several hours worth of questions to ask you. But the good news for our audience is that we will finish at 7 o'clock on the dot. So thank you again. We're now going to move on to Jay Self. Jay is a pediatric ophthalmologist at Southampton Eye Unit and also associate professor at the University of Southampton. His research interests include albinism, nystagmus, which he describes as wobbly eyes, and genetics. So over to you now. Thank you.
Everybody, welcome to my Gift of Sight lecture. Thank you for bearing with us, and apologies for the background. It's very odd sat in your bedroom in a jacket talking to a computer screen, but this is part of our new normal. So without further ado, I'm Jay Self. I'm a paediatric ophthalmologist and a paediatric eye researcher, and I work in Southampton. My particular focus of interest is conditions affecting children and young people, and I work very closely with Helena Lee, who I'm also representing today. So some key facts about children's eye diseases, which people often find quite surprising, are that 1 in 6 children actually have a visual impairment of some kind. Now, this might be something subtle, so it might affect one eye only. It might be something that only affects vision and reduces it such that children can't drive in the future. But these things are hugely important for lives which are going to go on for many, many, many years. When we're seeing children at 1, we're dealing with their eyes for the rest of their life. The important point is that children often have poor vision when they're young, and if we don't fix it by the time they're 7 or 8, they generally will never see well out of that eye or both eyes. So if you cover a child's eye with a patch early on in their life, really only for a week or so, they often will never see well out of that eye. The other interesting fact is that we're not a specialty that's dominated by a particular disease. Rare disease is by far the most common reason for blindness registration in children. So if you take all children who are registered as having a severe sight impairment, the commonest cause is individually very rare conditions, but when you combine them together, they become very common. The fourth interesting fact is that 1 in 3 patients in Southampton Eye Unit is in fact a child, although you may not see them because we squirrel them away around the back. 1 in 3 patients in Southampton Eye Unit is actually under the age of 18. So I wanted to talk about a few examples. We do a lot of research and a lot of conditions, the broad theme being that it's for young people and children. And as I say, most of my work is done in conjunction with Helen Lee, so I'm speaking on behalf of both of us today. So a few examples. The first example is a very common condition. So this is one of the conditions which makes up that 1 in 6 children, and it's squint and lazy eye. So this is where 2 eyes do not look in the same direction. The brain gets 2 different images, so it chooses one, ignores the other, and unless we get the brain to start listening to that other eye, the child will not see out of that eye after the age of 7 or 8. The treatment for this is patching. So we put a patch over the better eye to encourage the brain to look through the bad eye, or we sometimes use eye drops to do the same. But 50% of children, and there's about 250,000 children in the UK patching right now, 50% of them will fail this for a number of reasons, which you can imagine as a parent, a busy parent with lots of children, keeping a patch on a baby's eye for hours and hours each day is very, very difficult. So the solution to this that we see is working on ways to encourage that patching, where it's working with the child, it's working with the family, it's enforcing the importance of it, it's information, it's support, it's all sorts of different things. And people have tried lots of different interventions to try and get kids to wear their patches more. But my colleague Dan Osborne, who there's a picture of him on this screen, Dan Osborne is working really hard to try and see which package is the best and whether it suits everybody or whether we need bespoke ways to try and encourage patching use. So he is currently a pre-doctoral NIHR fellow, and he's applying for a PhD studentship, and his study is going to be looking at how we can increase this patching, not only locally but across a number of centres, and that's a study that he's going to be doing for a year or so, hopefully through this PhD studentship with NIHR. So that's one of the conditions which is really common. The study that he's doing is using things that we already do and looking at the best way of doing it. So it's very, very clinical end, doesn't involve a lab at any point, but has the potential to have huge implications for a very large number of patients for the rest of their lives. In contrast to that, example number 2 is dealing with a disorder which is relatively rare. So this is albinism. So this is a condition which causes loss of pigment in the body, so the body can't make pigment, and that has lots of implications for the eyes, meaning that people with albinism have significant visual impairment. As you can see from the 2 pictures at the top of the screen, sometimes albinism is very obvious, but importantly it's often not obvious. So lots of children go undiagnosed. Lots of treatments or early phase treatments that are being looked at really depend on the underlying cause, so we need to get to the bottom of what the cause is for an individual. And the third problem we have is that genetics are complicated. So the solution is to see lots of patients, take lots of samples, find out what they've got, find out what genetic changes can cause albinism, increase the yield in the NHS, so get people to actually test for it and really get to the bottom of diagnosis, and also make sure that this isn't just done in specialist centres like us, make sure it's done across the board. So the outputs that we've had for this are that we've worked on this for quite a number of years actually, and we've done lots of genetic studies finding out all the different types of causes for albinism. We've done lots of publications on that, we've written book chapters, we've run seminars, etc. We've also worked on NHS diagnostics, and I'm pleased to say we managed to get our panel of genes approved by the NHS. So now this is done, looking at all the genes properly across the UK and beyond, so worldwide as well. In line with that, we've developed a group of people interested in nystagmus and genetics across the UK called NUC. I'm not sure I remember what the acronym stands for. Oh, it's written on the slide there. But it's NUC, and that's a group of us who are really trying to push forward better care for people with nystagmus and albinism. Nystagmus being movement of the eyes, which is one of the features of albinism. At the moment, we're working on a care pathway, again with the idea of making sure that everything is not just done really well in big centres, but it's done everywhere, because most patients are seen in much smaller centres. So the third example is looking at the same condition. So this is albinism, and thinking beyond diagnosis. So diagnosis is really, really important. You need to know what somebody's got if you're ever going to be able to help them with anything. So the problem is that currently there are no treatments. So even if we get to the bottom of diagnosis, there's no approved drug therapies or surgeries to treat the actual cause of the condition. Most children are registered sight impaired. So although we've improved diagnostics hugely and patients really, really value that, we want to go to the next step or the next stage and make sure that we can try and do something about it. So the solutions here are laboratory research, again, taking samples from patients, going back to the lab, seeing if we can build models. So these are little cellular models, things in trays, in dishes, in which we can test some drug therapies which might work. It's also about then going on to looking at early phase studies in humans. And so the outputs we've had with this, the most notable is Helena's very large MRC grant, which is looking at a particular drug called L-DOPA, which is one of the pigment pathway parts. So it's one of the things that's missing in people with albinism, and that she's shown that this can work really, really well in labs. It can even work really, really well in animal models. And so she's just going forward now to look at early phase clinical trials. So that means testing it in humans in a small number over the next year or so. And this is a huge, enormous process to go through, involving lots of money and involving lots of different people helping and contributing, but she's doing a fabulous job with that. The other thing I wanted to mention in this rare disease treatment is related to these little pictures that you can see up here. You might be able to see these little pictures of tubes with funny coloured blobs at the bottom. These are essentially blobs of human cells where we've changed the genes and we've changed them in the way that we see in our patients. So these are the same spelling mistakes we see in patients, and we managed to change these cells so that they've got the same spelling mistakes. What that means is we have a mini patient in a tube. So when we're testing new drugs, we might have 100 or even 1,000 drugs to test. You can't do a clinical trial on each one of them because each trial would cost millions of pounds and take years and years to do. So what we can do in the lab is test them on our mini cell versions of patients, and we can do lots of them simultaneously really quickly. So a couple of weeks to perhaps test 100 or even 1,000 potential drug therapies. So developing these cell models is really key to it. And this is something we've done in the last couple of years as well. So those three examples are quite disparate. You can see that some of them are about very early phase lab work. So this is working with cells, working in the lab, doing complicated genetics, and some are right at the other end of the spectrum, very much patient focused, changing what we do on the shop floor on a day-to-day basis to try and improve outcomes. But the common theme again is that patients are at the centre of all of it. We make sure that everything we're doing is related to increasing patient care and improving patient care, and that it has a tangible way of doing that. We're not particularly focusing on extremely rare conditions which only affect 5 children every 10 years or so, because we're interested in improving things for the most patients we can. It also illustrates really that we're particularly focused on clinical work and lab work, so we appreciate both ends of that spectrum, and both can pay dividends, and often they work together, hence the lab to clinic link. So if I was to summarise what we do in our paediatric research in Southampton, the focuses that we've had and are still having going forward are on diagnosis. So we really fundamentally believe that to really get to the bottom of helping people with eye conditions, we need to know exactly what's going on, what the condition is. The second thing is to develop new treatments through the methods I've just described. And finally, and really importantly, we want to standardise care so that it's not just patients seen in 1 or 2 big centres in the world who get the best care, but actually this best care model is rolled out. So finally, just a quick thank you. This is a thank you to all of our funders. So we've had lots of funders over the years. I've given up doing a slide of all our collaborators because we have so many, both in-house, in the Vision Group, across the wider university, in other universities in the UK and worldwide. So I don't put that slide there, but I do thank our collaborators hugely for all their work. And finally, the most important people to thank are our patients, without whom we would be able to do no research at all. And their enthusiasm and their energy really keeps us going. So thank you to them. And thank you all for listening.
Thank you, Jay. That was excellent. It was a timely reminder to me of the sheer numbers involved in a paediatric department. We tend to think perhaps of eye diseases being the domain of the more elderly in society, and you have thoroughly quashed that. Thank you very much. So we go on to our final speaker of the evening. This is Professor Andrew Lotry, who is a consultant at Southampton Eye Unit and holds the chair of ophthalmology at the University of Southampton. Much of Andrew's work is about genetics. I'm sure he's going to at least mention that this evening as he walks us through some of his latest research, the clinical trials, and the impact this may make upon blinding diseases. Over to you, Andrew.
Hello, and thank you for allowing me to speak at this Beating Blindness biannual lecture from bench to bedside. My name's Andrew Lotry. I'm a professor of ophthalmology here at the University of Southampton. So our goals are to understand the common causes of blindness. We want to do this so we can develop better treatments for our patients. And we want to train the next generation of researchers so this work can continue. One of the commonest diseases we work on is age-related macular degeneration. It's the commonest cause of blindness in the developed world. And to better understand it, for the last 20 years we've studied the genetics of this condition. And as a result of this, we have come up with potential new therapy, which is replacing a faulty gene in the complement system using a new technology called gene therapy. And I'm pleased to report that this has led to clinical trials which are underway at the moment, and we eagerly await the results of this. If successful, this could lead to the first treatment for dry age-related macular degeneration, which is a huge unmet need. And this illustrates the benefits of studying a disease at the most fundamental level, and by understanding it, in this case through genetics, it has led to a potential treatment which is now in clinical trials. Indeed, with colleagues around the world collaborating, we've identified many genes that cause age-related macular degeneration. Indeed, when I started work in ophthalmology, no genes were known for this disease. Now we have 52 different genetic variants that can cause this disease. And we're starting to work through them to understand how this causes disease so we can get better treatments. I mentioned that one of our goals is to train the next generation of researchers, and I'd like to illustrate that through this photograph of two of our talented researchers who've come through our training programs. Dr. Rebekah Kay, we recruited from Liverpool. She was an academic clinical fellow with us and is now doing a PhD under my supervision, along with Dr. Jorn Lakowski. Miss Helena Lee was an academic clinical lecturer with me and is now won a highly prestigious MRC grant to develop her own independent research in pediatric eye disease. And she's been appointed as an associate professor in university and also as an honorary consultant at Southampton Eye Unit. And Helena has also started our first ever neuro-ophthalmology clinic. So I just wanted to make the point that as well as training the next generation of researchers, we've also attracted the brightest and the best, which is of benefit to our clinical service, such as by introducing a new clinical service, as Helena has done, that wouldn't otherwise been possible. And so both Helena and Rebekah have come from outside Hampshire to work here in Hampshire, and that's a benefit to our patients because of the skills they bring, as well as the research that they're doing. So Rebekah, as I mentioned, is doing a PhD, and she is using stem cells to study the function of T-span10 in the back of the eye. T-span10 is one of the genes that we've found to be associated with an increased risk of macular degeneration. And again, using powerful new technology, we can take cells that are undifferentiated and convert them to the cells that grow at the back of the eye that are otherwise very difficult to study, and these are called retinal pigment epithelium cells. And using an amazing technique that allows us to edit out a single gene what's called genome engineering, Rebekah's been able to remove this gene from these cells, and then we can study the effect of it not being there. One of the first things we've noticed is that the pigmentation in these cells is different, and they have less pigmentation than cells that have this gene in it. This suggests that pigmentation is important and may be important for development of macular degeneration. And we're studying this further, and this potentially could lead to new treatments. We think, similar to how we studied other genes that have led to clinical trials at the moment, that by understanding T-span 10's function, we might be able, in the long term, to find a drug to treat age-related macular degeneration. So these examples highlight why it's important to study diseases at a very basic level at the bench, to understand the mechanisms that then give us the insights we need to develop treatments. The other advantage of working both in the clinic and in the lab is that we can also gain insights from patients. And GiftAsight has helped us do this. It paid for a special retinal camera that cost over £100,000. And because of that support, this enabled us to win a £4 million Wellcome Trust grant. If you're a financial person, this equates to a 40-fold return in GiftAsight's investment, which is a very good return. But more importantly, it allowed us to win this really prestigious grant, the first time an ophthalmologist had ever won it, and allowed me to bring together a really talented group of the world's leading experts from all over the world to study macular degeneration in this collaborative award. And what we're doing is using artificial intelligence to understand AMD, that is, using computers to gain new insights. I'm showing a picture of a single retinal scan, and this scan has 1 million pieces of information in it. Because of our collaboration, we've been able to bring together over 500,000 scans like this, which is an incredible amount of information. And it is only by using computers that we can process this amount of data. And so by using artificial intelligence, we're developing a better classification system than a human grader can make. And we're coming up with new ways to predict who is at risk of progression from the early stages of disease to the later stages, which will help us in our clinical management. I'm showing here a picture that looks like a scan of a patient's eye as it develops macular degeneration. But actually, this is not a real scan. This is a scan made by a computer of what it thinks a human eye is. So we have taught the computer to understand what the retina looks like and how it looks when it ages. So this is an entirely artificial scan, but it tells us that computers are starting to understand the process of how macular degeneration develops. And we think this will allow us to find new ways to predict who will progress with age-related macular degeneration. This is a really exciting cutting-edge project. Also, as part of this project, we're using Hubble Space Telescope technology. We're using the technology called adaptive optics, which allows us to get very focused images of the back of the eye, and we can see individual cells that sense light, rods and cones at the back of the eye in a living human being. And I'm showing you some of the pictures from this. They're a little bit hard to interpret, but essentially what we're seeing for the first time is that inflammatory cells in the living eye congregate at the point that there is early macular degeneration. And we can see these cells called macrophages moving around over areas of damage. And we weren't aware of this before. It's a new insight. And it's because, again, we're using this cutting-edge technology that we can start to understand this aspect of macular degeneration. So moving a little bit further along the bench to bedside process, we also do lots of clinical trials. And I'll just mention one of these, and this was a trial to look at central serous retinopathy, which is another retinal condition. And I was awarded funding from the government organisation NIHR and the Medical Research Council to do a clinical trial which I led across 22 sites in the UK. This was testing a drug called eplerenone. And I know that my patients always want to know when we're going to get a new treatment. And just to say, it takes a long time to do this. It took 8 years of work for this trial. And we thought it important to test this drug in a rigorous way, as it was the commonest worldwide treatment. And so we did the first randomized controlled clinical trial and then communicated the results to patients. Now, unfortunately, this drug was not found to be effective, but that's an important result because it stops patients getting the treatment that could potentially be harmful. And it also illustrates that even though many doctors thought this was an important treatment, it turned out not to be effective when looked at in a rigorous manner. And that means that we can now start looking for different treatments to treat this condition and stop patients getting the wrong treatment. And that's why we have to do clinical trials. So I'd just like to share with you the video that we communicate with patients to show how we try to make sure our results are as accessible as possible.
Are you in the dark about how to treat chronic central serous chorioretinopathy? Eplerenone, developed for treating heart failure, is widely prescribed to manage CSCR, even though it can have rare but important side effects. But does it work? A British research team working across 22 NHS hospitals ran a clinical trial to find out. They randomly assigned 114 patients to drug or placebo and followed them for up to a year. CSCR causes fluid to accumulate under the retina. Many anatomical measures were taken. But the primary aim was how well the person could see. How well people could see was measured by how many letters they could read on the letter chart. Over 12 months, the placebo group showed a small improvement in vision, as CSCR can naturally come and go over time. And how did eplerenone do? Eplerenone showed the same small improvements over 12 months. So, eplerenone does not work for CSCR. All the other things we measured have shown the same thing. The findings are particularly credible because almost all patients attended almost all visits and engaged in the study as planned. So the study had more data than expected, and the researchers are really sure eplerenone does not work for CSCR. Hey, cheer up. At least this knowledge will stop eplerenone being used to treat CSCR to no avail, and future research can focus on finding a treatment that does work. And you can help. Watch out for future studies that you can sign up to as a health professional or patient.
So to summarise what I've been saying, we're using a wide variety of techniques to develop better treatments for a range of eye conditions. We study disease at a very basic level in the lab with stem cells and genetics. We use clinical data and the latest artificial intelligence science to study unbelievable amounts of information. And we do clinical trials to confirm results so patients get the right treatment. And this is our from bench to bedside approach. Gift of Sight is essential to all of this through its early support for our work. So thank you very much for listening, and I look forward to taking any questions. Thank you.
Thank you, Andrew. I've been listening to you speak now for 20 years, I think, and every time it becomes just a little bit more exciting. This is fantastic work, and I never thought I'd see you show a cartoon. Well done. That's a major step forward. We're now going to move on to the question and answer session, and I can bring up a few here. We'll just run through them quite quickly, I think, that we have rather a lot. So the first is about stem cells. So this is really you, Andrew, just to perhaps touch again on RPE and T-span 10, which is from Julie Ann, and asking us about what future do you see for stem cell technologies? Are you hopeful, and when will it all really happen?
Thank you. Well, I think stem cells are useful now to model disease in the laboratory, and because, as I've shown, we can recreate those cells at the back of the eye that we couldn't otherwise access to look at function and treatments. I think there are some, the retina is like a sandwich with 3 very integrated layers and then a support layer underneath it. So it's like a 4-layer sandwich. So technically it's possible to put back one of those layers, the very bottom layer, with stem cells now. But what isn't possible is the 3 layers above that all interconnect and connect through the optic nerve to the brain, to have them connect in a way that light can be sensed. So in terms of a whole eye replacement or replacing all of the retina, I think that's still a long way off. And it's important not to raise expectations that a stem cell treatment that will cure all layers of the eye that are damaged by various eye diseases is going to be, here in the next few years. That said, there are tremendous insights happening all the time. The ability to create from adult cells, say a skin cell, to reverse that adult cell and turn it back into a stem cell and then convert that stem cell into the tissues at the back of the eye. That's only been known for about the last 7 or 8 years, and it was an amazing, unexpected insight. So it's very hard. I think Niels Bohr said it's very hard to predict the future, and it's very hard to predict when stem cells will translate into a retinal transplantation in that replacing all the layers of the eye. But we are using these cells to advance our research at this stage, and that can, for example, help produce new drugs, which would be another perhaps more cost-effective way to treat lots of people.
Thanks, Andrew. I'm aware, of course, now that for research, people can grow sort of mini retinas to experiment on, can't they?
Yeah.
And perhaps I thought I'd ask you just to say a few words and then maybe ask Arjuna if that's the kind of thing that will help him in his work.
Yeah, so I think that's where, at the moment, stem cells are really helpful in the lab to test new treatments, to look at mechanisms of disease. And as I say, 1 layer of cells, the retinal pigment epithelium, there are clinical trials in terms of replacing that layer of cells. However, for most retinal degenerations, we also need to replace the rods and cones, the photoreceptors, and that isn't technically possible as yet. So I'll pass over to Arjuna for his comments.
Thank you, Andy. It's true that we can use these, RP cells, retinal pigment epithelial cells, which we can get, the stem cell derived from donors. So they are exactly the diseased RPE cells, and we can model them. We can get real insights into exactly what is going on in that monolayer, that carpet of cells, which I described in my cake-like cartoon, and also find out what's happening inside those cells. So Andrew and I collaborated with a group in Finland recently. We have a stem cell program where we published getting some new insights into a really rare eye condition called Salisbury's disease. And the insights from that all came from these donated tissues, which have been, turned into stem cells and then into stem cell derived RPE cells.
And if I could just say, it's very useful being married to a dermatologist, because she taught us how to do the skin biopsies from these patients, which allowed us to then isolate these stem cells. But the point is, you can, from an adult, you can take a skin biopsy And I still think it's alchemy that you can turn that into a skin cell into a retinal cell. And I'm grateful for the patients who helped us with that study.
Jay, can I just come to you? You talked about your patient in a test tube and the genetic modifications. Will stem cells help your research?
Yeah, good question. Unfortunately, my wife isn't a dermatologist, so less helpful to me. But no, I think for pediatric eye disease, the problem that we have most of the time is we're quite a few steps further back. What I mean by that is we don't even know which tissue it is, let alone which molecule or which cells which are being damaged. So for a lot of the conditions we look at, we're still trying to find out exactly what's going on, what causes the condition in a really basic level, and then we can kind of work towards which cell type is at fault, and then that theoretically could lead for some conditions to thinking about what we can do about those faulty cells, and one of the options is stem cells. But no, I think it's a broad answer. Generally, we're a number of steps further back in pediatrics because we deal with lots of rare conditions.
Okay, thank you very much. The next question is for me. Hello, Julianne, who asks, what do we think about the use of robots in eye surgery? And the answer is it has been done. A man called Rob McLaren up in Oxford used a robot to do an operation actually as the kind of work that Prof. Lotry does, to inject cells behind the retina to cure a very rare disease. And it was a surprisingly successful thing. For routine retinal surgery, the truth is it's very slow and very expensive. You have to rig up an enormous kind of jig from which the instruments go into the eye, and you have a patient rather anxious there under local anaesthetic. So for the moment, not happening routinely, but for some very precise techniques like injecting under the retina, then yes, I think it will come. Thank you. A question for you, Andrew Lotry from Bish, which is, since the original laser trials on AMD, has the morbidity of the disease changed? In other words, are we actually helping anybody?
Well, yeah, it's a great question. So I'm really pleased to say that because of research, the rates of registration of blindness have gone down over the last few years, and that's despite seeing an increase in the number of people with age-related macular degeneration. So over the last 20 years, clinical trials have developed treatments, particularly for wet macular degeneration, and they've had an amazing impact. And the first time in history, we're actually seeing registrable blindness decrease. And that, to me, is a real indication that we're on the right track in terms of developing treatments that, despite ever-increasing numbers of patients, we are, to some extent, beating blindness. There's obviously a lot more to do, but the data would say that we are having an impact. And actually, we've looked at this around Europe, and our success rates are actually the best in Europe. And I think that's a testimony to the NHS, where once a treatment is funded, patients can get access to it. Other countries like Germany, there are restrictions. You have to apply for funding after, say, a few injections for wet AMD, and that delay in getting approval leads to worse outcomes. So, a pat on the back for the NHS that both through clinical trials and through delivery, we're actually doing pretty well, particularly for wet macular degeneration.
I'll go into a question from Jennifer about what are the current treatments. I think you've covered wet disease and how well we're doing. When it comes to dry disease, I'm just picking out from other questions that have come through. And one is, this is to you, Arjuna, which is, do you honestly think, do you genuinely think we might be able to change AMD or change the retina through changes in our diet, or is the damage already done once it's there?
Well, thank you for that really excellent question. So, what we're understanding now is that, diet has a really big impact on sort of predisposing a retina that would otherwise age normally, healthily, to something that might become diseased. There's been a few studies, and this is now really new research I'm talking about, where they are starting to look at reversing the effects of bad diet. There is some evidence to show statins in mice, I have to say, has an effect. And one thing that's worth bearing in mind is that you have these genetic risk factors for AMD, and there's evidence to show that by having a good diet, you can even sort of ward off, if you like, the risks that are imparted by a bad set of high risk genes. So yes, the short answer is yes.
Thank you. So I think, Arjuna, we're kind of talking epigenetics, aren't we? The stuff that turns our genes on and off, or perhaps turns them up and down. And that's probably one of the most exciting parts of research at the moment, isn't it?
Absolutely.
On a similar note, I've got a question here asking, do you think that an unhealthy diet might contribute to other conditions, for example, glaucoma and cataract, not just AMD? And I'll ask the others to chip in as well. So does bad diet cause glaucoma and cataract?
So we know that in the developing world, malnutrition leads to cataracts. We are seeing that, as Arjuna said, that a Mediterranean diet, even if you have genetic risk, if you have a healthy diet, if you don't smoke, even if you have you've inherited the bad risk factors, you can still delay progression of your disease by switching to a more healthy diet. That's probably true for cataract as well. Glaucoma, I'm not so sure about, but we are seeing increasing environmental factors for glaucoma. Some other work that we've been involved in collaboration has shown that air pollution, for example, can increase your risk of developing glaucoma or macular degeneration. And that's very recent work. So we're increasingly seeing, even with genetic risk, you can modify the environment and get a better outcome.
Can I just come back to the inflammatory effects that were mentioned and the presence of these macrophages in the retina, and the extent to which diseases are perhaps inflammatory in origin? And of course, one thing I read recently is that I think there's an association between glaucoma and dental caries. So to what extent does the panel think that these diseases might just be manifestations of inflammation?
So what's really interesting about dental caries is that actually causes complement activation in your mouth. And complement, as we find, is a major risk factor for age-related macular degeneration. So again, these are interesting associations that hadn't been thought of before. So there's also an association between bad teeth and Alzheimer's disease, again, through this inflammatory link.
Yes, perhaps everybody goes off and starts flossing and using mouthwash, just to remind people that it is healthy to have bacteria in your mouth, and killing them all is not necessarily the answer.
Yeah. Can I chip in here, Andy?
Yeah, of course.
Yeah, no, I was just going to say that I think a lot of, if we zoom out a little bit, a lot of it is based on conditions which are due to cells which become sick through a number of different routes, then being susceptible to a disease which looks the same at the end. So there are types of glaucoma which are purely plumbing based, for which it's harder to imagine some of these environmental factors. But there are types of glaucoma, definitely cataract, which are due to cells which are susceptible to sickness of any type, which is why it seems like all of these disparate things add together to give you one condition. So it absolutely does make sense for certain types of glaucoma. And yes, I agree with the cataracts as well. I did an eye camp in India a few years ago, and the average age was 35, and that was not genetic based. So yeah, yes. Totally agree.
Jay, while I have you, we have a lot of questions, just a couple on the same theme. One is the L-DOPA being used to treat albinism the same as that used to treat Parkinson's?
Absolutely, yes, it is. And also it's the same as it's used in various dystonic movement disorders in children as well. So it's not a completely innocent drug, which means that there are a huge number of loops to go through. But yes, it is the same drug.
Thank you, Eileen, for that question. And a question from Wendy, which is, are patients with albinism more at risk of Parkinson's?
That's a good question. Not as far as we know. So there are a number of links. There's different types of albinism. So some produce less L-DOPA in one particular way, some don't. So it's a complicated thing. But as far as we know, there's no evidence for that at the moment.
Thank you very much. Just a few more rapid fire as we are running out of time. An opinion on taking supplements containing omega fatty acids plus vitamins C, E, and zinc and copper. Now, Prof, you might like to tell us what the ARED study said about the taking of fish oil type supplements.
Thank you. So there's been 2 large vitamin trials done in the United States, looking at different vitamin supplements, and they showed that a combination of vitamin C and E, copper and zinc, lutein and zeaxanthin reduce your risk of progression from the early to the late stages of macular degeneration by about 25%. Part of that study did look at omega acids as well and didn't find that they had any benefit in reducing progression. So you can purchase what are called the AREDS vitamins at several manufacturers, which have the same combination of vitamins that were in these studies. And they, as I say, in these clinical trials, they did show a significant effect in people who had moderate signs of AMD. In very early signs of AMD, they didn't really seem to show any effect, but at moderate to late, they did.
Thank you. A couple for Arjuna. A question from Hannah Gaston. Hello, Hannah. Does the type of dietary fat matter in the development of AMD in your mice?
So what I with the 10 minutes, it's very difficult to say that with the breadth of the study. So the diet we used has a very high fat content, but it also has a high sugar content. And this is exactly what you find in a Western diet. And the other thing that's worth noting is that on BBC One today at 9 o'clock, so this is beautifully timed, program on the effect of eating highly refined processed foods. And you can watch it all at 9 o'clock. So I'm not going to say anything else about it, but it's a beautiful link. And the link is that actually all these diets, high cholesterol, high fat, high sugar, has absolutely devastating effects. And there's a real visual feel to it when you watch that film at 9 o'clock. I've listened to his interviews beforehand, and it's going to be pretty spectacular.
Yes, perhaps you might just mention very briefly, perhaps, that the work of Weston Price, who was a dentist back in the 1930s, wasn't he? Who went around the world observing the fact that people eating very unrefined, non-Western diets didn't seem to get our diseases.
No, that's right. I mean, you know, it's always the case that studies have shown you have this Oriental or Mediterranean style diets, which has high fruit content, fresh nutrients, low in processed foods, hardly any refined grains. They are also associated with better eye health, better retinal health.
Yes. Yeah. Coming back to what Prof was saying about the fish oil supplements, I think it's fair to say, and Prof can chip in obviously, that there have been studies suggesting that eating fish is a good thing. Of course, a lot of Mediterranean countries do, but to come back to you, Arjuna, so if somebody said to you, so what is a Mediterranean diet, given that the Mediterranean is a pretty big place, what's your answer to that?
I don't think there's a set recipe, is all I can say. But I think our understanding of a Mediterranean diet is what it is. You know, it's kind of less red meat, less processed meat, fish, grains, fresh vegetables.
Yes, I mean, to come back to Weston Price, his point was it's not just what we are eating, it's what we're not eating. It's how one food displaces another. I think that was the theme, wasn't it, really? Yeah. Now, folks, I think, yes, the clock is against us. There are many questions come in, and we're going to have to leave some for another day. I have a number of announcements to make, but the first is to let the audience into a little secret, which is that when this kind of event is planned, the worry is that we might have what's called a slow audience, and therefore we have questions just in case. Thank you. I said to the organisers, we've never had a slow audience, so we won't have one tonight. So thank you for proving me right. A bit of housekeeping. I believe there will be a short poll on your screen, which we ask you to complete. That will take no more than 2 minutes. You might want to watch these lectures again, so you will, I believe, receive details from the mailing list. And also on the mailing list is a chance to watch the university's next event of a similar nature, which is a virtual Sailing on the Edge, the science behind the sport. Sounds fascinating. And that is on Wednesday, the 14th of July. You should receive information via your mailing list and you just sign up. So thank you all for attending, and I'm going to hand you over to Professor Lotry for his final thoughts. Andrew.
Thank you, Andy. I'd like to thank the audience for attending and also my co-speakers for their excellent talks. I hope this event has given you some indication of the breadth of eye research now underway at the University of Southampton and the progress we're making towards beating blindness. The gift of sight appeal has been instrumental in allowing us to do this work, so I also thank you for your support of the gift of sight appeal. I look forward to meeting you at our next event, hopefully in person, but until then, good night.